Good afternoon. My name is Audrey Scherrman. My research was a study of the migration of tumor cells that occur in the central nervous system. For this project, I worked under Dr. Li Yao in the Department of Biological Sciences. To begin, I wanted to touch on the central nervous system a bit and shed some light on how it would normally function in a healthy individual. The central nervous system is comprised of the brain and the spinal cord, and it is the regulation center for our body. So what that means is it controls your homeostasis, so how you regulate your temperature and heart rate. It's also responsible for your perception of the environment with your senses. It also coordinates your bodily movement and control, and then it is also the cognitive center, so it is responsible for your thoughts, your moods, and your memory. So when a tumor occurs in the central nervous system, it's going to start to interrupt these processes. Some of these disruptions can present as altered vision or loss of hearing, as well as aphasia. It can also disrupt your cognitive center, which results in an altered personality or even loss of memory. These tumors can be benign or malignant, but both can result in a decrease in quality of life and even in death. For example, around 60% of all malignant tumors reported are actually glioblastomas. Glioblastomas only have a 6.75-year survival rate, meaning they are fatal over 90% of the time. So therefore, it's very important for us to study these tumor types because there's not a whole lot of therapy options out there right now. A large part of how the tumors begin and start to invade our cells are as a product of where they're derived from. So neurons and glial cells are the two most common progenitors of tumor cells. Neurons are actually responsible for transmitting those electrical and chemical signals. And then the glial cells play more of a supportive role in that they surround the neurons and protect them. So to study migration, we want to be able to recreate the real-life environments that affect migration. We actually noticed that that's mostly in the cell membrane. The cell membrane mediates the interactions between a cell and its environment. That can be through protein interactions, so like the proteins on the cell membrane interacting with the extracellular matrix. That can also be done through membrane potentials, so changing ion gradients in and outside the cell. An actual interesting part of that is that the changing in ionic concentrations, that can actually result in an endogenous electric field. That can be seen in studies where they have studied wound repair. The cells are actually electrically guided to repair sites from the changes in ion concentrations. It's also been reported that cancer growth can create these electric fields because it's disrupting its environment and these gradients. Because of this, we decided to set up a two-part study. We want to investigate migration by mimicking that cell membrane environment, and we also want to see what happens when we apply electric stimulation as a result of the ionic concentration. So to begin, for the membrane-like environment, we used a type of cell called a PC12 cell. These cells are derived from the adrenal medullary of a rat brain, and they are neuronal in nature, meaning that they are like a neuron and can give off a neurotransmitter. For the second part of the study, for electric stimulation, we chose U87 cells. These are derived from a human glioblastoma tumor, which is a brain cancer cell that originated from a glial line, so that's supportive nature. Both of these cell types were cultured routinely, and we also fed neuron growth stimulation to the PC12 cells in order to give them those processes to study their migration with them. The study covered two parts. So for the cell membrane-like environment, we wanted to understand two conditions, and we did that as a coding technique. We took both collagen and polylysine and coated a 6-well plate, and then saw how the cells would respond and grow on top of that coating. Collagen is important because it is found in our body, so most importantly, like the extracellular matrix and some cell membranes. And then polylysine is more of a chemical. And what that does is increase the electrostatic interactions between a cell and a surface it adheres to. So it's increasing that negative and positive charge to make it kind of more of a membrane acting. And then for the electric stimulation study, we actually created a specific tissue culture dish where we made channels. We did that by applying slides and creating kind of this dip in the center. And then from there, we kind of made it more of a galvanized structure. And what that means is, on the top here, that is a typical galvanic chamber where a salt bridge runs in the middle to direct the current from right to left. What we did is modified that, and we actually introduced two salt bridges. So we ran from the Steinberg solution, which is a salt solution so it can run the electrons through. We ran it through our cell culture, through the center, and then back out to create that chamber. And then afterwards, for our results of the membrane study, if you look on the left, section A, that's going to indicate the polylysine coating. You can see here that there is some migration behavior still occurring. The cells are still moving across that axis. If you look on the right, that is actually the collagen coating. And you can see that there is a lot of migration here. They are moving all over the axes. And from this table, if you look on the left, that's an indication of speed. We see that collagen has a higher speed. And it also has a greater distance traveled if you look on the right. And then as the results of our electric stimulation, on the left, section A is actually 1 hour of control, so there was no stimulation applied. And then on the right, that is section B, where the stimulus was applied for 1 hour. From these trends, you might be able to infer that there is a directionality occurring. And then as well on here, if you look on the left, the direction seems to shift more upwards or go in the opposite way. And then if you look on the right, it looks like the stimulus might have an effect on the speed as well. To conclude the results, I did just want to state that although we can notice the trends in the migration of the stimulus, there's not enough statistical significance for us to for sure say that they would be migrating towards an anode or a cathode. Additionally, further exploration of the stimulus strength, how many volts we're using, and how long we're exposing the cells could yield better results. Unfortunately for this project, resources were a limitation and we were not able to do those replications. And then moving on to the cell membrane, for investigating this environment, we did show that the collagen is a much better coating surface or membrane-like environment than the polylysine. However, just because we do see a better migratory behavior there, that's not telling us that collagen, we should use collagen more than polylysine. In fact, collagen, since it is a protein, it can interfere in a further downstream application where we would want to extract the protein and perform like a DNA or an RNA analysis. Polylysine is a good chemical component for that because we do show the migration behavior and it would not interfere with a protein interaction. So all in all, both coating methods do have uses for future applications. And then I did just want to remind everyone that it's important to study these migration behaviors because it links us to cancer metastasis and future research and future therapy goals. And with that, I want to give you my references I cited. I would also like to thank Kay and Bree for their additional support. And then this concludes my presentation, so I'm opening the floor for questions. Well, cell migration is the key component of how a tumor cell metastasizes and invades its environment. So if you study how it's moving and what factors are addressing the movement, that can better lead us to understand a potential therapy method for cancer. Like, for example, could electric stimulation guide a tumor growth in your brain? If we can do that instead of doing an invasive surgery or instead of chemotherapy that doesn't typically work, that could be a better therapy outcome. So migration habits are a direct correlation to cancer metastasis. I did kind of touch on that as well. We would want to do further replications and studying different voltages, different durations, and length of time it's affected. There's actually already been studies on this in a more of a matrix style or three-dimensional culture, to where there has been results found to where the glioblastoma cells can migrate with an electric field. We can use this to apply to a bunch of other studies that don't even have to do with cancer metastasis, like Parkinson's disease, cells that occur in the brain. If we are doing an electric stimulation study, we can better determine a treatment outcome.